for the opportunity to speak today. It's always a privilege to come to the family meetings and meet the parents and hear about their stories and learn what everyone else is doing. It's always been a good opportunity for me to frame the importance of what we're doing in the lab, and it's a source of motivation. So what I'll be doing is telling you more about our work, which is from the perspective of the molecular and the cellular abnormalities in AHC, which I hope will complement what you heard from Dr. McCarty's lecture on what happens at the tissue level in the brain and the whole organism. And I think it's important to realize that we need to stitch together all of that information, going from the gene to the pump, to what happens at the cell level, to the tissue level in the brain, to the organism, before we can really have a good, firm grasp of the mechanism underlying AHC. And that's really one of the best ways that we can be sure that we target the right thing when it comes to therapy. So I'd like to begin with a few slides as background. And some of you may have heard me give this sort of primer on ATP1A3 before. And so if you've heard it before, this will be review. If you haven't heard it before, hopefully you'll find it helpful. But I think there's some fundamental aspects of the ATP1A3 protein that you should understand before I can try to persuade you with our data. First of all, ATP1A3 encodes a molecular pump for sodium and potassium ions. It's a protein that is floating in a lipid bilayer, which is the cell membrane, which is basically a sea of lipid molecules that are arranged in a certain way. And this pump basically spans the entire thickness of the cell membrane, so it can actually have access to the outside world and the inside world at the same time. This schematic shows you the function of the pump. The pump does two main things. It moves potassium ions from the outside of the cell to the inside of the cell in exchange for sodium ions, which are pumped out from inside the cell to the outside. This is an energy requiring process. The energy is derived from the splitting of a molecule called ATP, and that provides a little spark of metabolic energy that turns the crank on the pump. And for every cycle of pumping, 2 potassium ions, and by the way, K is the atomic symbol for potassium, don't get confused. Potassium ions, 2 potassium ions are pumped in exchange for 3 sodium ions. And again, sodium, the atomic symbol is Na. And that's why we call it the NAK ATPase, sodium potassium ATPase. Now that's an interesting pump because it creates this asymmetry of ionic charge. If you're moving 2 positive ions into the cell, but removing 3, you have an imbalance. And that imbalance is best illustrated by this clever rendition of a Drake music video showing the 2 potassium ions coming in and the 3 sodiums moving out, and then he kicks ATP to give the energy. If this is all you remember about this part of the lecture, that's fine. This is the pump in action. So the reason that's important is because that asymmetry of charge, which is less elegantly exhibited by my graphic here, leaves a net negative charge inside the cell. And you can measure that with a voltmeter, and you can, in virtually every cell, the inside of the cell is negative as opposed to the outside. And at rest, we call that the membrane potential. Potential is a fancy word for voltage. So there's a small, tiny voltage across the cell. And again, it gets there because if you had a sea of positive and negative charges on one side of a membrane and another sea of positive charges on the other side, and at the beginning they were all equal. So there's no net imbalance. You throw a voltmeter into this, measuring from this side, and you move one little positive charge too many over onto the other side, this side becomes negative relative to the other side. So this is an important point because the ATP1A3 pump sets the resting membrane potential, or it sets parameters and conditions and contributes to the resting membrane potential. So that's point number one. The resting membrane potential relies on the action of this pump. If the pump wasn't working, you might expect an abnormality in the resting potential, and that would create other problems. A resting potential and the ionic gradients created by this pump are essential for firing what are called action potentials. Those are the impulses that travel along nerve fibers that signal the things that we do in our nervous system. And so again, if the pump isn't working, one might expect abnormalities in the ability to generate action potentials. So we've been modeling this disease in human neurons that we derive from specialized cells called induced pluripotent stem cells. These are so-called stem cells, but they're derived from a body cell, not from an embryo. And they are now much easier to create than they used to be. And through a collaboration with Kevin S. at Vanderbilt, his lab has stockpiled a number of induced pluripotent stem cell lines, which I'll call IPS cell lines from here on in, has stockpiled those from patients with AHC, representing the most common mutations and a few less common mutations. The idea behind this technology is you can take a cell from an individual patient, turn it into this IPS cell type, and then from that IPS cell, they can be now induced to become any other cell in the body. They become neurons, they become heart cells, they become anything. And this is the paradigm that we have now allows us to now investigate cellular properties of a disease for which we normally would not have access to those cells. We can't get brain tissue from someone with AHC, living brain tissue. And so we create it in the laboratory. So here's the list of the mutations. We have a bank of mutations now that have been collected from patients that Kathy Swoboda has seen, Kevin S. has seen, some others are in the list too. This is not an updated list, but just gives me a talking point to say that we have representative samples from all the top three most common mutations listed here, and some unusual ones. And I'll be talking a little bit about the three of these mutations in my talk today. So this is a picture of some induced pluripotent stem cells that have been stained with certain types of antibody stains to represent their properties. The next step is we actually take those cells and put them through a cascade of exposures in the laboratory to turn them into neurons. This is some pictures of early days where the cells are just starting to convert from being IPS cells to neuron progenitors to neurons, and then even more extensively. This process takes a few weeks every time we do it, so it's a slow process. And I have a name down here, Christine Simmons, who's partly cut off, but Christine is in the audience there. She's trying to wave. Here's some more elegant pictures. Here we actually stained for ATP1A3. We wanted to make sure the neurons had the gene we were interested in studying, and they do. And then our first few experiments, the first few lines that we were able to study in the lab all had the mutation G947R. That's the third most common mutation. It was one of the first patients we encountered that Dr. S's lab was able to make stem cells on. And fortunately, we had both a female and a male with the same mutation, and we had different lines, different cell lines from the female patients. So in all, we had 3 different cell lines that we were able to work with, and from each cell line, we made multiple batches of neurons. And the reason we're doing this in multitude is simply to improve the rigor of the experiment so that we're not basing all our conclusions on one patient and one clone of that patient and one batch of neurons. We do things repetitively over and over to make sure that we're seeing something consistent. So now what we've mainly done with these neurons is to ask the question, what is the fundamental defect that we see in the activity of the pump that we can measure in the neurons and other cellular properties? And so the way we have approached this is we do single cell measurements using a sophisticated type of electrophysiology measurement where we can actually detect tiny, very tiny electrical current flowing in the cell. We do this one cell at a time. Here's a picture of a cell that exhibits a red color. Attached to it is this, what looks like a giant probe. This is actually a very tiny probe. It's just blown up in the microscope. But this is a patch pipette, so-called, that allows us to make these measurements. And we can make a whole host of measurements. I'll be showing you things that are representative of the electrical current that comes from the pumping action of ATP1A3. We call that pump current. We can measure the resting potential of the cell, which depends on ATP1A3. And then we can look at action potentials, which are those pulses of electricity that are fired in neurons. So just to begin with, we thought the most fundamental thing that we should be trying to find is any evidence, direct evidence, that ATP1A3 is functional or not functional. That was just a fundamental question that was not completely appreciated and had never been studied in human neurons from someone with AHC. And so in this representation of our data, this is a measurement that we made in AHC neurons from a healthy person. These should be normal. And what we see is there's very little activity in the beginning, and that's because we've deprived the cell of potassium. So there's nothing for the pump to do. There's no potassium for it to grab onto and pump. But then we suddenly flood the chamber where the cell is being recorded from with a very high level of potassium. And suddenly the pump can go to work. It pumps potassium aggressively into the cell in exchange for sodium. And for every cycle where there's this unbalance of charge, there's a net positive charge leaving the cell. And that creates a little bit of electrical charge that we can measure. This is a very small charge. It's on the order of a billionth of an ampere. Nothing is going to curl your hair, but we can measure it. And it's there for the duration of the exposure of the cell to the potassium. When we stop putting that potassium in there, we actually have a decay of the current back to baseline. And then if we repeat the same experiment, but include a compound called wabane, which blocks the pump, we actually blunt the effect. And so this suggests that we're seeing some kind of wabane-sensitive pump activity in these cells. Now that's a normal neuron. What about an AHC neuron? We see less pumping. There's a slower rate of rise, although I'm not sure that's held up consistently as an effect. And certainly the amplitude is blunted. There's less pumping activity. We can look at many, many cells and then average the data and display it in this manner, where we have a range of measurements that we've made, and this line in the middle is the average of all those measurements. Here we have simply the gray symbols represent the control healthy neurons, and the red or pink symbols represent the AHC neurons. And in every case, with a total current averaged over several cells, we see there's less pump activity, less pump current that is measured. Now we can look at the two components, the wabane sensitive part, which we think is likely predominantly coming from ATP1A3 in a neuron, with some contamination by ATP1A1, which is present in virtually every cell. And the wabane sensitive current is also considerably depressed. So we feel like this is good evidence that there's, and direct evidence that there's low pump activity in these human AHC neurons. Now there's some other things we can do with that, and some additional measurements that we can make. If in fact the ion pump isn't working well, that means that it won't be able to accumulate in the neuron the amount of potassium that normally would be there. Now, normally this action of the pump keeps the interior of the neuron low in sodium concentration, but high in potassium concentration. Well, we can actually infer the potassium concentration by measuring something called the equilibrium potential. Now, this is really advanced stuff here, but basically this is a measurement that basically infers the ratio of the external or the outside potassium concentration relative to the inside potassium concentration. We make this measurement easily in the neurons, and then we can back calculate what the concentration of potassium is inside the cell. Normally we think that's pretty high, 130 to 140 millimolar. In our experiments with the normal healthy neurons, we got a value of 127, pretty much what we would expect. In the AHC neurons, 46, profoundly low. And again, this is now a little bit of indirect evidence that the pump isn't working as well as it should. We can also use these data knowing that the membrane potential is a sum of potassium and sodium and chloride conductances in the membrane. And we can actually back calculate what the resting membrane potential should be. The calculated resting potential for a control neuron is minus 59. For the AHC neuron, it's minus 40, almost minus 46. So very much less negative. We actually measured it, and on average, we found very good agreement. The measured resting potential for the control neurons is around minus 58, whereas for the AHC neurons, it's minus 45. And here is some graphic illustration of those measurements of resting potential. Again, we looked at multiple clones from one of the patients with the G947R. Here's the control neurons. What's plotted here is resting membrane potential, abbreviated RMP, a value of around minus 58. And then the AHC patient's neurons are around minus 48. We looked at multiple rounds of neurons that we derived from that patient. We looked at 2 different independent cell lines from that patient and saw a very consistent result. We then looked at the male patient with the same mutation, same trend in the data, and here's just a table illustrating all the measurements we made, hundreds of measurements, well, several dozen measurements, and consistently the AHC neurons had a less negative resting membrane potential. Again, all very consistent with the observation that the pump is not pumping, the ion gradients are disturbed, and the resting potential is not what it should be. So how does this translate then? If the resting potential is abnormal, how does that affect the ability of the neuron to generate an electrical impulse, the so-called action potential? And that, again, we can do with this single cell measurement. We can actually excite the neuron electrically and watch whether it fires an action potential or not. And what you see in this first panel is a little spike of electricity that goes up and then recovers, and then another one, and then not so much after that. And that's pretty typical. The more current we inject into the neuron, the more spikes we see. And we just simply count the spikes and correlate that with the amount of current that's injected into the neuron. And normally we get this nice plot like you see here. For the control or healthy neurons, you can see that we get a couple of action potentials at maximum, and then it plateaus. For the AHC neurons, we usually just get 1. So this suggests that there's a blunting of the electrical excitability of those neurons. They're not responding as well as they should. Now, it turns out this experiment was done letting the neurons exist at their native natural resting membrane potential, which I showed you previously was abnormal. It was less negative than it should be. If we then correct for this, artificially making the resting membrane potential a fixed value, in this case, minus 80 millivolts, not that physiologic, but we pushed it really negative. When we do that, control neurons and AHC neurons look exactly the same. So this impaired ability to generate action potentials is sort of a functional defect, all due to the impaired pumping activity and the impaired resting membrane potential. We then figured out that this, in fact, this impairment in the ability to generate action potentials was simply due to the fact that certain other elements in the neurons, specifically sodium channels, that have to activate and allow sodium into the cell to cause the action potential to fire. Again, this is sophisticated stuff, I apologize. That was also abnormal when we measured the cells at the resting membrane potential that they had, but not if we changed it to minus 80 millivolts. Everything was the same. So again, we think this is a functional defect. This is just more of the same data. So again, everything kind of tracks back to the pump isn't working. That creates an abnormal ion gradient in the cell and a less negative resting membrane potential, and that impairs the neuron's ability to fire. Now, we've looked at some other neurons as well. I'll show you just some preliminary data here on D801N, which is the most common mutation associated with AHC. And here again, we measured the resting membrane potential. We're still working on measuring the pump current, by the way, but we measured resting membrane potential in control neurons and D801N neurons, and what we see is the same trend, a significantly less negative resting potential. So far, that's been a consistent observation for two of the most common mutations. Now, I want to stop for a moment and just give you a broad perspective of what we're trying to learn here. First of all, the patients are with AHC have a mutation in ATP1A3, but they also have another copy of ATP1A3 that's perfectly normal. The genetic term for this is heterozygosity. They have a normal copy and they have a mutant copy, and that's sufficient to have the disease. The question now is this disease simply, is the mechanism here because there's a missing copy or a non-functional copy of the gene, or is the mutant copy somehow poisoning the other normal copy of the gene? And that difference is often referred to as, on one end, haploinsufficiency, which is simply a lack of one normal copy of the gene, but with one normal copy still existing, versus a dominant negative effect where the mutant copy somehow doesn't work, but it also poisons the activity of the normal copy. So just to illustrate that, here, for example, is, say, somebody's two copies of ATP1A3. The normal healthy copy is in blue, and the mutated copy is in red with this little star after it. Haploinsufficiency is best illustrated by just imagining that the mutant copy is just not there. It's not there, and this person exists only because they have half the normal copy of the gene, and that's just not sufficient for normal activity. Now, our data doesn't really reconcile that very well. I'll come back to this point in a moment. Dominant negative is a different story. So again, we'll start with a normal copy and a mutant copy, but in this case, there's something about the non-functional or dysfunctional copy that poisons the normal copy, so that neither works very well at all. And the reason this is pretty important, and I think we've heard a little bit about gene therapy this morning from Dr. McCarty's lab, is if the idea is dominant, if the idea is haploinsufficiency, then supplying more blue ATP1A3 should solve the problem. If it's a dominant negative mechanism, then providing more normal ATP1A3, well, the question is, how much more do you need? And this, we don't know. So that's something that has to be reconciled. Now, the reason I bring this up is because the common mutations are all single amino acid changes in the protein. The protein is likely to be made and exist in some fashion in the cell. And so we can't really easily differentiate between these two mechanisms. That said, if there was a mutation in ATP1A3 that undeniably wiped out the complete protein, where it didn't exist at all, and we could be sure about that, then haploinsufficiency might be a more likely mechanism. Now, this is not to say that you can't have both, haploinsufficiency with some mutations and dominant negative effects with some others, but we like to simplify things and imagine everything is simple and clean and perfect. So we're looking for one reason here to move on. So in some cases, in some genetic diseases, there are mutations that completely destroy one copy of the gene. The protein's really never made, it doesn't exist in the cell, and so we really don't have to worry about a dominant negative effect. It's almost always going to be haploinsufficiency as the primary mechanism. So those types of mutations we don't see in ATP1A3. I think still to date, there's not been an obvious truncating mutation identified in this disease, like a premature stop codon or an obvious frame shift mutation. They seem to not be there. And so we actually looked very hard at the list of mutations, and one particular mutation stuck out at us, which we thought might be one of these truncating mutations. This was a so-called Splice Site mutation. Here's a little clinical vignette, which is a little less important, but the mutation itself didn't directly affect the protein. It was in a nucleotide position outside of the part of the gene that codes for the protein, ATP1A3. This is illustrated here. This is a blow-up of some DNA sequence from exon 18 of ATP1A3. The lowercase letters in black are the intron part of the gene, which is the part that doesn't code for protein. The capital letters in blue are the coding part of that exon, and that will eventually translate into a protein component of the gene. And then we have more intron sequence. The mutation in this case sits right outside of the protein coding region. It's this red T. Normally that should be a T, but in this mutation it became a C. And the reason this is important is that is an area that we call a splice site. A splice site is a junction between an exon, which are the protein coding regions, and an intron, which is the non-coding region. And virtually all genes have a mixture of exons and introns. And what the cell does is it reads that, translates it into transcribes it into RNA, and then cuts out all the introns and stitches together the exons so you can make a nice protein. Well, they depend on these little signals called splice sites to do that properly. And the first two letters of an intron, the G and the T, are really essential. In this case, one of those essential bases is a C. It's not correct. And so we imagine this might actually create a missplicing event, which could end up causing some kind of truncating effect on the protein, which was sort of what we were looking for, any evidence of truncation that might be a clear sign that haploinsufficiency was the primary mechanism in this disease. So to really examine that in more detail, Kevin S.'s lab made iPS cells from someone with this splice site mutation. Then Christine Simmons in my lab grew up neurons, made neurons from there, and then isolated RNA from those neurons, and then determined, was that RNA correctly spliced or not? And so she determined that it was not correctly spliced in all cases. In fact, again, there's a mutation copy and there's a normal copy. When we do this experiment with just the healthy neurons, we see a single copy that's correctly spliced. In the case of the mutation, we see one band at a lower level that's correctly spliced, and then we see another band that's misspliced. And the missplicing in this case causes not a truncation, as it turns out, but rather an insertion of some extra sequence that came from the intron that was supposed to be removed. It wasn't completely removed, it was partially removed, and what was left just created a 22 amino acid insertion into the gene. All right, so this is not a truncating mutation. This does not provide us that much clarity at the sequence level that this, in fact, is going to be haploinsufficiency. But we had to do one more thing, which was, did the protein get made in a neuron? What I showed you here was actually the RNA analyzed. This is now the question is the protein made? So to do that, Christine looked at the protein level using a technique called Western blotting or immunoblotting, where we have an antibody against ATP1A3, and if it's present on an electrophoretically separated panel of proteins from the cells, so-called Western blot, we can detect it. So as illustrated in these healthy neurons, we see this big, beautiful band of protein that is representative of ATP1A3. And in the neurons from the patient with the Splice Site mutation, we see a band of the same molecular weight, but at lower density, but nothing bigger. We do not see this extra band which would represent the protein with the insertion. That we don't see. It's not there. Probably it's not stable in the protein. It gets degraded in some way, but it's not present. And the ratio of the two remaining proteins is illustrated here. We have about a little more than half the amount of protein in the Splice Site mutation line as we do with the control line. And this is not the case for other ATP1A3 mutations, as illustrated here. This is Western blot data from control neurons compared to neurons made from iPS cells with the mutation G947R, and they're equal. There's no diminished effect. So we think this is a small piece of evidence suggesting that at least in the Splice Site mutation, there is only one copy of ATP1A3. So in this case, there can't be a dominant negative effect. So do we see now, we go back to the physiology experiments, do we see the same impairment in the pump activity, and do we see the same degree of abnormal resting potential? And so here is some summary data showing the recordings of pump current made in neurons from the Splice Site mutation cell lines compared to a healthy control. We see the same pattern. It's diminished overall, and then if we just look at Wabane sensitive current, we see a significant diminution in the pump current. Resting potential also abnormal, and here we looked at two completely different types of neurons. We looked at cortical excitatory neurons, which is what we had been working with. We see this less negative resting potential there, and we also looked at so-called inhibitory neurons or interneurons. Dr. McCarty mentioned that in his talk, and we see the same effect. So there's a depolarized or a less negative resting potential in those neurons as well. And this, again, would predict, as we saw before, an impaired ability to maintain the proper ion gradients that you need to generate action potentials. So by our prediction, this would also suggest perhaps that the ability of these neurons to fire action potentials might be blunted. We have to prove that. We are still doing experiments with these mutations, and more on that will come later. So I think this is some evidence suggesting that maybe these disorders are really haploinsufficiency rather than dominant negative, and that may impact how we think about treating them going forward. Now, I'm going to leave. I think that's, well, it's news. It's news. Okay, Kathy's excited. I'm excited too, you just can't tell. I'm excited inside. This is a good segue because I want to switch gears and briefly talk about two other things that we've been doing in our lab. From the earliest phases, we've been very interested in asking the question, could we find a drug or a drug-like chemical compound that would activate the remaining ATP1A3 pump? Could we goose it into action somehow? And did a drug exist already that we didn't realize had that capability? Or could we create a strategy to screen thousands of compounds for the ability of something to activate the ATP1A3? Right now, to our knowledge, no drug can activate this pump. So how do we find that? So we've done a couple of things. First of all, we developed a cellular assay system. It doesn't involve neurons. It involves another type of cell that we can use to deliver ATP1A3 into an assay system. And we can actually measure the pump activity reasonably well. And we use that pump for a few years when I was still at Vanderbilt to screen all the known approved drugs. There's about 1,300 approved drugs here in the US and also in Europe. And we went through, I think, 2 different libraries of those approved drugs from different sources. Did this a couple of times. And I will say, I think we've conclusively determined that there is no current drug that can activate ATP1A3. We've looked at some other libraries of drug-like compounds, and again, we're disappointed. Those libraries might not have been the best libraries to screen, but to suffice it that that phase, I think, is pretty conclusive right now. There's no approved drugs that we think activate ATP1A3. The other strategy, which became an issue when I moved to Northwestern, or an opportunity, was there was a computational chemist named Rama Misra who had a system whereby he could model a protein in the computer and then simulate a chemical compound, a small chemical compound, docking with that protein. And he could actually, he did this for us with the structure, the known structure of ATP1A1 that we then simulated ATP1A3. So we took the structure that's known for ATP1A1, threaded the sequence of ATP1A3 human into that, remodeled it, and have what's called a homology model of the protein. And then using that, he systematically did a docking experiment in the computer where he took each and every one of 10 million chemicals and docked them to the structure. Now, first of all, we sat down with him for a long time and we looked at the structure of the protein that he had derived, and we said, you know, there's some very common mutations that are clustered around this position in the protein at 801. And so he studied that for a long time, and we met, and he said, look, there's a nice little binding pocket right there. There's, I don't have my glasses on. But here's aspartic acid 801 right there. This is the position where D801N is mutated. And there's right next to it, there's this really juicy little pocket that he said, let me try to dock into that and see what we find. So he found about 50 different chemical compounds in this library of 10 million that seemed to fit beautifully into that. Here's some renderings that he made for us. Maybe hard to see, but in this little pocket, there are these little chemical stick figures that are stuck in there. Now that's just a screening experiment. The next thing we had to do, again, work done by Christine Simmons in my lab, was to actually decide experimentally if any of these 50 or so compounds did anything to the pump activity. So we did it 2 ways. We first of all used a screen that is kind of crude, but it does inform us as to whether the cells have active sodium-potassium ATPase activity. But we can render them susceptible to death when we expose the cells to wabane, and then we can pre-treat the cells with a compound we think activates ATP1A3 and test them again. And so I realize this is confusing, but normally what we saw was that normal cells didn't die. Sorry, this is cell survival. Anyway, the bottom line is we found a few compounds that seem to protect the cells from wabane-induced death, which suggested maybe some of those are actually activating the pump. But then we actually went directly to the pump assay, the thallium assay that I talked about before, and some of these looked like they were not having much of an effect, but perhaps 2 of them were activating the pump to some extent. Now, this was hopeful, but certainly not a definitive answer. We've gone on to do some new experiments, and I don't know that this is going to end up being significant at all. More work has to be done, but this is just to give you a sense for what we've been trying to do, finding small molecules that might be pump activators. This is not over yet, but I think I don't have any definitive answers for you today. Last but not least, we've been using a mouse model that was developed in Denmark by the lab of Karen Leakey-Hartmann in collaboration with Thomas Holm. And when I asked Karen if I could have the mouse model to do some experiments on, she said, sure, and gave me Thomas as well. So we got 2 mammals for the price of 1. But Thomas has been in my lab for the last couple of years, thankfully supported in part by the AHC Foundation. And he's actually recreated a colony of these animals at Northwestern. These animals have consistent features that were described in some recent papers. They have some baseline neurological impairments, including ataxia, attention deficit, hyperactivity, and they have a slightly reduced seizure threshold, although if you breed them onto different genetic backgrounds, that completely goes away. That's something Thomas determined in my lab this last year. But importantly, they have stress-induced attacks of weakness and dystonia, not unlike the D801N mouse that Dr. McCarty showed us this morning. And so that assay that we use is to expose the animals to cold water, to lower their core body temperature to 23 degrees Celsius. It's about room temperature. And then we take the mice out and we observe them and videotape them and then monitor their recovery over time. Now, a couple things I think are really important to mention here. First of all, Thomas realized that the body weight of the mouse correlated with how fast they recovered. And we realized that the animals weren't all cooling to the same extent. So now, as he does these assays in a very rigorous way, he pokes the animals with a temperature probe and monitors their temperature throughout. And we leave them in the water just at the moment they're 23 degrees in core temperature, we take them out. So that is really